Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled AI-based Antibody Therapeutics Developability Assessment and its Engineering. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank GenScript, who developed the content for this presentation. GenScript is the leading contract research organization in the world providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. Now I'd like to introduce our speaker for today's event. Carter Howe received his PhD in Control Science and Engineering from Zhejiang University of Technology. He is now a bioinformatics senior scientist at GenScript and mainly takes charge of the research and development of antibody-related applications. He has published 20+ papers in his research fields in international journals such as the European Journal of Pharmaceutical Sciences and Information Science. But now, without further ado, I'd like to go ahead and hand the presentation over to our speaker, Carter Howe. Carter, you may begin when you're ready.
Okay, hello everybody here. Thanks, Ryan. And thanks all of you to attend the webinar. I'm Carter Howe from GenScript. I'm the senior bioinformatics scientist. Okay, and today I'm going to share the recent progress on antibody engineering at GenScript. And especially on the AI-based developability assessment of the therapeutic antibody and its engineering. And the presentation contains following parts. The antibody therapeutic. Okay, let's move. Sorry, hold on. Okay. The presentation contains following parts: the antibody therapeutic development trends as the background, and the introduction of our immunoinformatics platform, and followed by the AI-based tools for the antibody therapeutic development. And at the end of the presentation, there will be the Q&A part. Okay. Let's now move on to the first part, the antibody therapeutic development trends. As we all know, the therapeutic antibody is a very, very important class of the therapeutics, and it grows fast. As shown here, there were several antibody therapeutics that got approved every year since 2010. And in this figure, you can see along with the years, and the number of the approved therapeutic antibodies is increasing. And in just past 2023, there are 16 first approved therapeutic antibodies. And there are nearly about 200 antibody therapeutics are available in total. While the development of the therapeutic antibody is always not that easy, most of the therapeutic antibody development are time-consuming with extremely high cost in the traditional wet lab-based methods. And in some of the parts, the search scope is limited due to the library size. For example, the humanization, since that we know we have to make the library first when doing the humanization or binding affinity maturation with the wet lab-based experiments. Well, fortunately, with the popular application of AI in biology field, It gives us the chances to solve many problems in the development of the therapeutic antibody, and with more and with rapid development cycle and cost effective. And in the part that with the limited search region, with the powerful computing ability of the computers, we can enlarge the library size to solve those questions. For example, the limited library size. Okay, and so there exists about 82 AI-related biotech companies which pay attention on the protein or antibody drug discovery, and more than 60 of them are just established in the last 5 years. And so AI is more and more important and popular in the biotech companies. And the AI and the antibody therapeutic development is also no exception. It becomes popular and popular. Okay, so next I'm going to introduce the AI applications in GenScript, the immunoinformatics platform. Here I list the general process of the development of a reagent or therapeutic antibody in GenScript. Starting from the antigen, and we go through with the animal immunization, sequencing of the antibodies, developability assessment, antibody reformation and production, and end with the qualified products delivered to our customers. Okay, today we will mainly focus on the developability assessment and the antibody engineering parts. And also, we have other tools to be briefly introduced. And then now let's move to the antigen selection and the preparation part. Okay, as you know, antigen is always the first step of the antibody development. And GenScript have our own internally used antigen analysis packages. And here I just briefly show you the actual prediction tool, which is mainly for efficiently animal immunization to get the animal source antibodies. And we regard the actual prediction problem as a classification problem to identify whether the given peptide is active or not. And here we plot the evaluation indicators for measuring the performance of the classification model in the figure. For the true positive and the true negative, the higher the better. And for the false negative and the false positive, the lower the better. Okay, and the performance of GenScript aptope prediction tool is plotted in blue, and we can see that our tools perform better than the comparison method, the aptope, which is published just in 2021. Again, and now with the aptope analyzed, next step is to express the antigen. And actually, no matter what kind of the proteins to be expressed, for example, peptide, small proteins and large proteins, maybe just like the antibody, the DNA sequence is essential for determining the expression level of the target protein. For example, the GC contents would obviously affect the expression and should be optimized. And in our newest AI-based codon optimization method, Zhuque performs excellent in optimizing the codon to increase the expression level. And we take the previous codon optimization method, GenSmart, to make the comparison. The above two figures here shows the Zhuque significantly improves the performance no matter in human, mouse, or rabbit species. And especially for those difficult to express IgG antibodies with the GenSmart, the Zhuque significantly improves the expression level. And below here, I list the significance of the codon optimization method and especially for the stations. Narrow codon optimization tool has been cited by more than 8,000 works in their publications. Okay, here, after the expression, animal immunization, ELISA testing, and NGS sequencing followed with the antigen get prepared. And we also have some internal tools for analyzing those sequencing results for making sure on our sequence, the sequences are all correct. And with those sequence, the antibody at hand, and we're going to do the developability assessment, including basic sequence annotations and about the PDM, aggregation, and the degradation site analysis, and also the immunogenicity with the viscosity risk assessments. Okay, and here is antibody chain domain annotation tool for letting the customer know basic information of antibody on sequence level. The system will separate the full chain to several parts, including the signal peptide, and tag, linker, B regions, C regions, and mark them with different colors. And we also evaluate the FR regions and CDR regions. And if the specific region is not satisfied with the evaluation criteria, the back flag will be marked. Just like here, all of those CDRs and FRs are good, so here marked with a good label. And also other informations like species, chain isotype, mutations are also provided in this analysis report. Okay, and GenScript also provide bispecific antibody-related services, and there are nearly 400 kinds of the bispecific antibody structures in our knowledge database. And the structure of a given bispecific antibody can be smartly identified with the BSAB structure annotation system. Each sequence will be split into several parts, and the chain ratio with the reference structures are also reported. Things like that here, we call this sequence mode for letting the users to know how your sequence is composed. And if the queried bispecific antibody is not identified in our database, for example, like this test case, we will return the sequence mode like here, and also the similar, the most similar one in our database, and also the corresponding related informations will be also reported here. And in our next in-house programs for developability assessment are for aggregation and degradations in a PDM size prediction program. Okay, here for the aggregation, we compute the spatial aggregation prompt score for each residues and identify whether the residue is a potential aggregation site or not. The upper right here, this picture is a model of 3D structures of an antibody in a mesh model with the SAP scores colored. The higher the SAP score is, the darker the red color is, and the lower the SAP score is, the darker the blue color is. And in this figure, we plot the SAP scores for each amino acid in this figure. The x-axis is the amino acid number for the heavy and light chains. And the y-axis here is the SAP score. And we set a cutoff here for identifying the whether this site is potential aggregation site or not. Once above this score, and those sites will be identified as the potential aggregation sites. And well, all of those scores below the certain cutoff, it will be reported as not aggregation sites. And in this test case, we just show you guys for just like here, the red regions. Those regions are to be predicted as the aggregation site regions. And for the degradation, we mainly consider the asparagine, the degradation, and the Masonia oxidation. And with only those few kinds of the features as the input to the machine learning models, and we can achieve a prediction accuracy equals to 94%, and the corresponding AUC score equals 0.96, and which means that a high prediction accuracy of our developed prediction model. And also for PTM, we use the protein BERT model as a base model, and with the use of specified PTM database for fine-tuning to get different PTM prediction models for each considered PTM type. Here in our immunoinformatic service platform, we consider those kind of the PTM types, the hydroxylation and methylation, glycylation, and phosphorylation. And all of the test results shows here that our prediction model achieves over 0.9 in terms of the AUC score here. And actually, viscosity and immunogenicity are other important factors to be considered when applying the therapeutic antibodies into bodies. And if we could get those kind of the viscosities and immunogenicity information of the candidate therapeutic antibodies once the sequences has been designed, and then we can make the corresponding decisions for choosing which candidate to be produced and further validated. Thus, both of the time and money could be saved, right? And even we can make corresponding reformations of those candidates to treat off the developability indicators. Definitely, we have developed our own method for predicting the viscosity and immunogenicity. Here, we show the pipeline for viscosity prediction. It is actually a large language model-based machine learning method. Our method starts with only the antibody heavy and light chains. And those chains was input to the Proti5 this model to extract the embedding as the features to represent the input sequences. And Proti5 is a large language model, pre-trained large language model. And we got those extracted embeddings as the features. These features, the dimension of these features is really high for the random forest like the machine learning based methods. So we have to do the dimension reduction. And in here we use the kernel PCA for do this dimension reduction. And with the compressed features obtained, and we got we input those features to the random forest classifier to get the predicted viscosity values. And as of the concentration at 150 milligram per milliliter, the viscosity value above and below 30 cP would be classified as high and low respectively. And this work has published on the European Journal of Pharmaceutical Sciences in early 2024. And here we show the performance comparison between several published sort methods and our proposed methods on the AB40 dataset. And then let's say this table. We listed the Sharma model. This is an experience-based formula to measure the viscosity. And here, these three methods, the SCM-related methods. SCM refers to the spatial chart map. These methods are developed from the Ping-Kwang Lai's research group. And the other one is the PIFNet, which is developed by Pfizer. Okay, and from those kind of the evaluation indicators, our Prote5RF. This model proposed by us achieves the highest indicators on all of those evaluation indicators, which means the Prote5RF performs best. Okay, and let's say here those figures, they are plotted for the different methods. And here this is our predictive method. The x-axis are all for the predicted viscosity value from different prediction methods, and the y-axis are all the experimental measured viscosity values. And the better the performance of the prediction method is, those points will come into a line here, which reflects the PCC value here. And as we can say, our Prote5RF achieves the highest PCC value, like 0.9. Okay, and this means that our proposed method performs best on the AB40 dataset. And next, we also take the AB21, this test dataset for comparing with our method with the Pfizer's methods. Those methods are trained. These models are trained by the LOCV method, which means the Liu-1 cross-validation. And on this test dataset, the test results show that our method is comparable to Pfizer. Okay, and here we also take some existing commercialized the antibodies, antibody therapeutics for measuring its viscosity, just like listed at here. The comparison method is from University of Michigan. They just published their methods in 2024. Yes, and the test results shows here. We list the experimental measured viscosity value, like in this column. And here is the GenScript predicted values, like here. And for predicted results, they just gave that these antibodies with high viscosity or low viscosity, but our GenScript prediction method gives the specific viscosity values here. And as we can see, the test results shows our GenScript prediction methods achieves 100% corrections accuracy, while the just achieves 70%. And similar to the viscosity prediction methods, our proposed immunogenicity prediction methods also takes a pre-trained large language model as feature extractor. And while this extractor, the large language model is Antibody, and with those extracted features, with the use of AutoGlone as the classification model trainer for getting the immunogenicity prediction model. And finally, we can get the predicted immunogenicity. We use the 88% as the immunogenicity indicators. With the 88% that's higher than 2%, and we will classify this antibody as high immunogenicity. And with the 88% below 2%, we will classify this antibody as the low immunogenicity risk. And this work also has published in early of 2024. Both of the papers are available online, and you can get it for more details if you are interested. And here we take the only previous existing method, which predicts the 88% as the immunogenicity indicators for comparison. It was published in 2020, It's called PITHA. Yes, and in terms of the AUC and PRCs, our ABMU prate, this is on our proposed method, achieve the highest indicator values. And above those two figures, and we plot the ROC curves and the PRC curves here. Both of those indicators, the higher, the better. And the consensus with the conclusion that we just mentioned, GenScript prediction model performs best in all of those indicators. And as all of the currently implemented developability assessment indicators in our immunoinformatics service platform has been introduced here, and I want to bring a good news to our customer or potential customers. It is that all of those developability assessment indicators can be obtained from our TurboCho Smart Quote online ordering system. This system will be released at the end of June. It's coming soon, yes. If you have interest in knowing the developability indicators of your design antibody, you can then try it for free, yes. Okay, and with those developability indicators at hand, one question you may want to ask is that could you suggest me how to do the modification? Yes, so next I'm going to introduce some antibody engineering services at GenScript. These services mainly for the humanization, canonization, felinization, and the binding affinity maturations. Okay. First one, the monoclonal antibody humanization services. There are mainly three kinds of the humanization strategies on the market. The CDR-grafted antibodies, and the chimeric antibodies, and the fully humanized antibodies. Well, the fully humanization method always expensive and may encounter the patent restrictions, while the chimeric antibody has considerably high HAMA risk. Only the CDR grafting method is much more popular with low cost and fast delivery. We have two ways for implementing the humanization. First one is the traditional bioinformatics relational design and AI-based design. Okay, those methods are all on the basis of CDR grafting and our own developed humanist evaluation models. Here, this slide shows two rabbit monoclonal antibody humanization cases. We show the BLI testing, binding affinity testing of the chimeric antibody and the humanized antibodies. Here, just like the binding affinity before humanization, in this case, it's just about 1.77. And the binding affinity after humanization, it comes, the value comes much smaller, which means the binding affinity is increased. And for this case, before the humanization, we tested the chimeric antibody. It is the rabbit human chimeric antibody. The binding affinity is just like 9.28, while the humanized antibody, it comes lower. The value is lower, and which means the binding affinity is increased. And so we can draw a conclusion that our humanization could have the chance to increase the binding affinity with a high success rate. Besides the humanization, GenScript also pay attention on pet care. We know that many people take dogs or cats as pets with them. So pet health is also very important to them, and there also is the need for animal antibody engineering. And we have also developed the corresponding animal-related services. such as monoclonal antibody canonization and felinisation. Here I show two cases we did before for rabbit monoclonal antibody canonization on the left side and the felinisation on the right side. Similar to the previous humanization, we show the pictures of the SPR binding affinity testing of the dog, the rabbit dog chimeric antibody and the rabbit cat chimeric antibody and the corresponding canonized antibody and the felinized antibody. Let's first see the canonization one. Before the canonization, the chimeric, the binding affinity of the chimeric antibody, it is the 2.03. While after the canonization, the binding affinity is yes, actually, as you can see, it becomes smaller. Yes, that says the binding affinity has been increased. And also for the felinization, before felinization, the chimeric antibody, the binding affinity of this, it is the 7.25. While after the felinization, the binding affinity score is decreased to 2.64, which means the binding affinity is increased. Which also we can draw this conclusion that our canonization or felinization strategies, it works and with the successfulness, and it also has the chance to increase the binding affinity. And we also have summarized the heterolization methods, including humanization, canonization, and felinization. We integrate them into a platform we called YAB externalization service platform. We summarize this method as a scientific paper and submit to this journal, and it is now under review. And I believe that it will come to see you soon once the review process has been finished. Okay. So next one, another important aspect of the antibody engineering is the binding affinity maturation. Yes, sure. Here we list a case for doing the binding affinity maturation, which shows the compared to the wild type antibody here. Most of our modified antibodies have a binding affinity increase, especially for the mAbs 6, 7, 8. While, as you can see, the increase, yeah, it is limited. But we are still working on this with the recent popular large language models and to train a new model for the binding affinity predictions. Yes. And the newly developed method is now on wet lab testing. And except the large language model AI-based antibody binding affinity maturation, the generative AI-based multi-species humanization, and also the multimodal evolution-based antibody therapeutic development reformation methods are also coming soon. Let's say how it works. And last but not least, I'd like to introduce the TurboCHO antibody expression platform. Currently, we only take 5 business days for high throughput expression and 10 business days for gram level expression, which is significantly faster than other providers, which always taking 6 to 8 weeks. Yes. And our platform received great feedback for its high yield, and we can even achieve gram level production for transient expression. And meanwhile, our prices are very competitive. And for more details on TurboCHO, please feel free to reach out to our GenScript sales team. Here that's all what I want to share today. And any questions, please?
Well, thank you very much for this very insightful presentation. As we move into the Q&A session, I'd like to remind our audience to keep sending their questions or comments in using the questions window for the Q&A portion. And if you're attending via GoToWebinar, if you're watching on LinkedIn Live events, feel free to post your questions in the comments section. So I've already received some questions from the audience, though, so we'll get ourselves started with those. The very first question that I have for you states that for PTM site prediction, if you identify a specific site as a potential PTM site, how would you make further modification? Further, they want to know, do you have any specific rules, or is it depending on the experience?
Okay, thanks this audience for proposing this question. Actually, since that PTM, it is more professional related to the biology and the biochemical scientist. Since I'm a bioinformatics scientist, so we would like to solve those kind of the PTM predictions in the bioinformatics way, and also solve those PTM problems in the bioinformatics way. Generally, we will make those mutations and then to predict the mutant sites will it be the PTM still be PTM sites or not? And if it is still predicted as the PTM sites, and then we make the further mutations and then do another round of the evaluations. Yes, that's the general methods for solving those kind of the problems. Well, the PTM, it is not a single problem to be solved. Since that we know many aspects should be under consideration of the antibody developability. So it is actually a trade-off between all of those developability assessment indicators. We should take all of the indicators as a whole and make those mutations and to predict that the PTMs, the degradations, and the aggregations, and so any other indicators like the immunogenicity or viscosity. Then all of those indicators come to satisfy the level, and then we know, oh, okay, this antibody is okay for next wet lab experiments validation and the next step for production and anything else. Yeah, that's all. Thank you.
Very good. Thank you so much for your insight on that question. The next one here is regarding overall accuracy of the platform. Someone's wondering, is it accurate enough to guide the reformation of the given antibody?
Okay, we also thank this audience for proposing this question. I believe that I have introduced the accuracy, the performance of each prediction methods, such as like the immunogenicity prediction and the viscosity prediction. The accuracy is good on their test data set. Yes, but if it is can be used for guiding the reformation of the given antibody, I just can say it is just a reference information for you, and we can take this information to eliminate some candidates that has the lowest probability to be the therapeutic antibody. Yes, and I believe that with the accumulation of the available data sets, yes, the performance of our prediction tools will get improved further. Yeah, thank you.
Perfect. Thank you so much. Now, the next question I have here is curious if you have any experience in producing or predicting antibody sequence using AI, and if so, how good are they comparing to the immunization generated sequence?
Okay, let me say where this question is. Do you have any experience in producing the antibody sequencing in this model? Do you have any experience in producing antibody sequencing? So good. You can immunize to generate. Oh, I'm sorry, we didn't do this before. We just predict those developabilities where we do not directly to generate antibody sequence. Well, it is also a very interesting project for us to do in the future. Yes, thank you.
Wonderful, thank you. The next question I have here then is curious, according to your platform, could the reformation change the original function of the antibody, and if so, to what degree?
Okay, as for the function, we think the antibody function is the most important function of the antibody is to bind the antigen, right? Yes, as you can say, the reformation could indeed to change the binding affinity to some degree, just like in the humanization or the canonization or felinization. The original antibody with the high binding affinity to the antigen, but when you do the humanization or canonization, the binding affinity has decreased or totally lost. This is really common. Yes, so it would be affect the antibody functions. Well, that's what's the meaning of our work, the bioinformatics or the AI to do the humanization or other reformations. We could to eliminate those affections in the computing way. Yes, thank you.
Right, no, thank you very much. Next, someone's wondering, in regards to aggregation prediction value of the residues, does the method compute only for isolated residues or provides for an average value for a stretch of residues?
Residues. Only isolated. That's the SAP score for each residues. We first calculate the SAP score of each atom in this residue, and then got the sum of the SAP scores of each atom in this residue. So it is the sum, well, not the average.
Okay, wonderful, thank you. The next question here is the affinity prediction technique you developed universal for all antigens, or does it require specific adaptations for different antigens?
It is universal. We can predict all kinds of the antigens and the antibodies. It's just you can provide the antigens and antibodies. We can make the fully saturation mutation in the cellular regions to find to measure the delta G of this mutation. And if the delta G decreased, then we think the binding affinity will be increased.
All right, thank you. Now, our next question here asks, can this platform be used to identify the antibody with lower affinity?
Used to identify the antibody with lower affinity? I'm not very clear about this question. What do you mean by identify?
Sure. So our audience member, perhaps you can restate the question. Of course, you will have the chance to follow up with our speaker afterwards. I've got another question for you, though, in the meantime, Carter. Someone's curious for aggregation prediction. If the region where the aggregation may occur is the functional region, how can it be optimized?
Okay. This question is similar to the PDMs. For those aggregations, if it occurs in the functional region, it mainly affects the binding affinity. That we can optimize it by the general mutation methods and then to evaluate the aggregations for the site to measure whether it is still the aggregation site or not, and then do another round of the mutations and another round of the evaluations until the PDM, till the aggregation sites has to be predicted as non-aggregation sites. Yes, thank you.
Yes, thank you very much for that question and for all of the questions today, but we have reached the end of our time here today. For those interested in hearing more from GenScript, you can register for their next webinar, Accelerating Cell and Gene Therapy Success with cGMP, Gene Editing Components and Regulatory Compliance, which is scheduled for Friday, June 12th. I've also sent a link in the chat box and for our LinkedIn event members in our comment box. Now, if we couldn't attend to your questions, though, do know that the team at GenScript will follow up with you, or if you have further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared links to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. And for those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event, gain access to features such as saving, as well as viewing the recording and more. I would now like to please ask you to join me in thanking our speaker for his time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you, guys. Thank you, Ray. Bye-bye.